Imagine a single-celled amoeba proteus, just floating around in a freshwater pond. Based on the fundamental laws of physics, water should be rushing into this cell so fast that it swells up and explodes like a water balloon attached to a fire hydrant.
Right, but it doesn't pop.
Yeah, exactly. It doesn't. And why is that? Well, welcome to the deep dive. Today, we're exploring the invisible borders that make life possible for you, me, and that tiny amoeba.
It is such a fascinating topic.
It really is. We are looking at how cells govern what gets in and out, and how those exact same microscopic rules scale all the way up to explain the actual mechanics of how you will take your very next breath.
There's this really elegant continuity in biology. I mean, the physical problem of keeping the outside out and the inside in it doesn't change, whether you are a single amoeba or, a complex mammal made of trillions of cells.
It's basically the same rule book.
Exactly.
Yeah.
At every single level, life is just this active, constant struggle against equilibrium.
Okay, let's unpack this. Before we can figure out how our amoeba survives or how our lungs function, we have to look at the architecture of the biological border itself.
The cell membrane.
Right, because the cell membrane isn't just a static wall holding the cytoplasm in.
Not at all, no. It is a highly dynamic structure, and it's primarily made of a phospholipid bilayer. Structurally, you have a molecule that is, it's thermodynamically conflicted.
Thermodynamically conflicted. I like that. What does that actually mean?
So the phosphate head of the molecule is hydrophilic, meaning it interacts easily with water, but it's attached to two fatty acid tails that are intensely hydrophobic.
They completely hate water.
Right, they repel it. So when you put thousands of these into an aqueous, watery environment, they spontaneously self-assemble.
Just because it's energetically favorable to do so, right?
Yeah.
Like the water-repelling tails tuck themselves inside to hide from the water?
Precisely.
Yeah.
Leaving the hydrophilic heads facing the fluid on the inside and the outside of the cell. And, for a long time, the exact nature of this arrangement was heavily debated by scientists.
Yeah, I was reading about that.
It wasn't until 1972 that Singer and Nicholson proposed the Fluid Mosaic Model. That model proved that this bilayer isn't a rigid shell at all.
It's constantly moving.
Yeah, it behaves more like a two-dimensional liquid, like a sea of lipids. And embedded within this fluid sea are various proteins, cholesterol molecules, and glycoproteins just floating around.
So instead of a static brick wall, it's more like a highly organized, crowded mosh pit.
A mosh pit, yeah. That's a great way to picture it.
Right. The lipids, they're constantly shifting and flowing, while the security guards, which are the proteins, are weaving their way through the mass, basically deciding who gets to pass through.
And that fluid nature is crucial for cell function. Cholesterol molecules actually tuck between the hydrophobic tails to modulate that fluidity.
So it doesn't get too stiff or too loose.
Exactly. It prevents the membrane from becoming too rigid in cold temperatures or, too permeable in high heat. Meanwhile, the glycoproteins act as molecular signatures, allowing cells to recognize each other.
The history behind how we actually confirm this bilayer even existed is so wild. I mean, in 1925, two Dutch scientists, Gorder and Grendel, they set out to prove it by extracting lipids from human red blood cells.
Yes, the famous red blood cell experiment.
Right. They measured the surface area of the extracted lipids and compared it to the surface area of the cells. The math showed a perfect 2-to-1 ratio, neatly proving the bilayer.
It would have been this perfect triumph of the scientific method, except they made two massive methodological errors.
Wait, really? What did they do?
First, they used acetone for the extraction, which isn't fully effective, so they accidentally left about a third of the lipids behind.
Oh, wow. That's a big miss.
Huge. And second, they calculated the surface area of the red blood cells, assuming they were perfect spheres. They completely missed that they are actually biconcave discs.
Which have a totally different surface area. So they massively underestimated the amount of lipid, and they massively underestimated the surface area of the cell.
Yes, but by pure mathematical serendipity, their two massive errors perfectly canceled each other out to give them the exact correct ratio.
That is hilarious, just failing upward in science.
It's a great reminder of the messiness of scientific discovery. But, their conclusion ultimately held up. Now, having this highly specific oily barrier creates an immediate problem.
How do you actually move molecular traffic across it?
Exactly.
Because cells need a constant influx of oxygen and nutrients and a constant outflow of metabolic waste. It seems like the rules of entry depend entirely on how badly the cell needs the material.
Well, it depends on the physical properties of the molecule and the concentration gradient. That's the foundation of passive transport.
Right. No energy required.
Zero. Molecules possess innate kinetic energy, causing them to move randomly. Over time, this leads to a net movement from an area of higher concentration to an area of lower concentration.
So for small, uncharged molecules like oxygen or carbon dioxide, the membrane is essentially invisible to them.
Right, they undergo simple diffusion. They just slip right through the hydrophobic core without any assistance at all.
But if the core of this bilayer is intensely hydrophobic, a charged ion like sodium or like a massive polar molecule like glucose, that isn't going to just slip through.
No, they would be physically repelled by those fatty acid tails. They are completely blocked.
So how do they get in?
That is where the proteins in our fluid mosaic come in. They facilitate the diffusion. Some proteins form dedicated channels with hydrophilic interiors, which basically creates a tunnel for specific ions.
And other ones are carriers, right?
Yeah, carrier proteins bind to a molecule, undergo a conformational change, a shape change, and release it on the other side. But critically, as long as the molecule is moving down its concentration gradient, it is still passive. The cell expends zero metabolic energy.
I like to think of passive transport as just rolling a boulder down a hill. But cells are constantly manipulating their internal environments.
Yeah.
They stockpile nutrients that are already highly concentrated inside.
And they push out waste into environments that are already saturated.
Right. So if passive transport is rolling a boulder down a hill, this is like physically pushing it back up the hill.
Exactly. And that requires active transport. Moving substances against the gradient costs energy, specifically in the form of ATP.
Adenosine triphosphate.
Right. The cell uses an enzyme called ATPase to hydrolyze ATP, which breaks the terminal phosphate bond.
And breaking that bond releases energy.
Yes. The energy released from snapping that bond forces the carrier protein to physically change its shape, actively pumping the molecule across the membrane.
The vulnerability of this mechanism is staggering, though. Like, if you look at a poison like cyanide, its lethality comes entirely from shutting this specific process down.
Cyanide is a perfect, albeit grim, illustration of how reliant we are on active transport.
What exactly does it do?
Well, cyanide binds to an enzyme complex in the mitochondria, completely halting the electron transport chain. Oxidative phosphorylation stops, meaning the cell can no longer produce ATP.
So taking out ATP is literally cutting the power to the cell's escalators.
That's a great analogy. The sodium-potassium pumps fail, the cell loses its ionic balance, it depolarizes, and it dies almost immediately.
Wow. And active transport isn't just limited to individual molecules either.
No, not at all. When a macrophage, like a white blood cell, needs to engulf a whole bacterium, it relies on bulk transport.
Right, endocytosis. For solids, it's phagocytosis, and for liquids, it's pinocytosis.
Exactly. The membrane itself folds inward, encapsulates the target, and pinches off to form a vesicle. And pushing large materials out via exocytosis works in reverse. Both are highly active, ATP-intensive processes.
Okay, here's where it gets really interesting. We've talked about solids and gases, but the most crucial, everywhere, all-at-once substance moving across these membranes is water.
Oh, absolutely. Water is fundamental.
And water operates under its own special rulebook, a specialized form of diffusion called osmosis, which brings us back to our amoeba.
Osmosis governs the net movement of free water molecules down a water potential gradient across a partially permeable membrane. The key concept here isn't just the sheer amount of water, but the amount of free water.
Let's break that down for everyone. When we talk about free water, we are talking about water molecules that aren't, physically tied up doing something else.
Exactly. When you dissolve a solute, like sodium chloride, into water, the polar water molecules cluster around the sodium and chloride ions. They form these hydration shells.
So those water molecules are busy. They are now bound.
Right. They have less kinetic energy, and they're less free to move around. So a highly concentrated, salty solution actually has a lower concentration of free water molecules compared to a dilute solution.
Got it.
Water will naturally diffuse toward the area with a higher solute concentration to equalize the free energy.
This brings us back to the danger facing an animal cell. If a human red blood cell ends up in a hypotonic environment, meaning the solution outside is pure water and very dilute, the free water potential is high.
So water rushes into the cell.
Right. And since the animal cell membrane is just a thin layer of lipids, it acts like a jelly-filled balloon. It swells up and eventually leases or bursts.
And if you put it in a hypertonic, salty solution, the water rushes out, leaving the cell severely shriveled.
But plant cells are different. They don't just pop.
No, plant cells evolved a structural workaround to this physical reality. They possess a rigid cellulose cell wall on the exterior of the delicate plasma membrane.
So when water rushes into a plant cell by osmosis, the cell volume increases, but the wall prevents it from bursting.
Exactly. The water just pushes the membrane tightly against the cellulose wall. That outward hydrostatic pressure is called turgor.
Turgor pressure, that's what keeps them standing up, right?
Fundamentally, yes. It gives non-woody plants their structural integrity. It keeps stems upright and leaves expansive to catch sunlight.
And conversely, if water leaves,
In a hypertonic environment, water leaves the plant cell. The membrane peels away from the cell wall, a process called plasmolysis, and the plant wilts.
wait, I have a question about this. If plant cells are under so much turgor pressure already, and water constantly seeks to balance out, why do plants need a continuous supply of water from the soil? Like, why do we have to water them?
That's a great question. It's because a plant is not a closed system. The atmosphere outside is generally far less humid than the inside of a leaf.
Oh, so it's constantly leaking water into the air.
Exactly, through transpiration. As water evaporates from the leaves, it creates a negative pressure that actually pulls more water up from the roots.
So if the soil dries out and becomes hypertonic because of a drought, the whole gradient reverses.
Right, the plant loses its turgor, and the biological machinery just grinds to a halt.
That structural wall is a brilliant adaptation, but it circles us back to the amoeba from the very beginning.
The freshwater amoeba.
Yeah, it lives in fresh water. It doesn't have a rigid cellulose wall. It just has that delicate lipid membrane. It is functionally a leaky boat in an ocean of pure water. So how does it bail the water out to avoid popping?
It employs a specialized organelle called a contractile vacuole. It is a masterpiece of cellular engineering that utilizes both active transport and osmosis.
So it burns energy.
Yes, the amoeba burns ATP to actively pump ions into the specialized internal vacuole.
Okay, wait. By packing the vacuole with ions, it's deliberately creating a localized hypertonic environment inside itself.
You've got it. It's tying up the free water inside the vacuole.
Oh, that is so smart.
By lowering the water potential inside the vacuole, the free water that was constantly entering the cell from the pond naturally flows into the vacuole instead of swelling up the main cytoplasm.
And then what? It just spits it out.
Pretty much. Once the vacuole fills, it moves to the cell membrane, contracts, and forcefully squirts the water back out into the environment.
It's actively manipulating physics to survive. I love that. But, as incredibly adapted as the amoeba is, it has a luxury that you and I do not. It is microscopic.
Very true.
A single cell can rely on simple diffusion for gas exchange. Even a tiny multicellular aquatic hydra, like 15 millimeters long, can just let oxygen diffuse directly across its skin.
But an organism of our size faces a really critical geometric problem. As an object grows, its volume increases cubically, while its surface area only increases squarely.
So our surface area to volume ratio drops off a cliff.
Yes. Humans have a massive volume of metabolically demanding tissue, but a relatively tiny outer surface area. If we relied on oxygen diffusing through our skin, it would take years for a single molecule of oxygen to reach our internal organs.
We would asphyxiate long before the oxygen ever arrived. So how do complex organisms cheat the limits of diffusion?
It all comes down to the mathematics of Fick's law.
Let's talk about Fick's law.
Fick's law of diffusion dictates the rate at which particles move across a barrier. The formula states that the rate is directly proportional to the surface area multiplied by the difference in concentration and inversely proportional to the thickness of the barrier.
Okay, so to maximize the speed of diffusion, you need three things, right? A massive surface area, a steep difference in concentration between the two sides, and a barrier that is as physically thin as possible.
Exactly. And the mammalian respiratory system is an evolutionary masterclass in optimizing Fick's law. The trachea branches into bronchi, then bronchioles, and they terminate in millions of microscopic air sacs called alveoli.
The surface area math here is just staggering to me. If you were to unfold all the alveoli in a typical human lung and lay them out flat, they would cover roughly 40 to 75 square meters.
It is an immense amount of space.
So what does this all mean? We basically folded up half a tennis court of incredibly thin, wet tissue and shoved it into our chest cavity just to hack Fick's law.
That's exactly what we did. That massive expansion solves the surface area variable in the numerator of Fick's law. And for the denominator, the barrier thickness, the walls of the alveoli are constructed from squamous epithelium.
Which is incredibly thin.
It is exactly one cell thick. We are talking measuring between 0.5 and 1.5 micrometers. The diffusion distance is practically non-existent.
And to maintain the steep concentration gradient, our blood is constantly flowing. The pulmonary circulation is pumping oxygen-depleted, carbon dioxide-rich blood right up against those thin alveolar walls all day long.
Right. As soon as oxygen diffuses into the blood, it is instantly swept away by red blood cells. That ensures the oxygen concentration in the blood never builds up enough to slow down the incoming diffusion.
But the structural optimization of the lungs is useless without a mechanism to constantly refresh the air inside those sacs, right? You need continuous ventilation.
You need the actual mechanics of breathing. And it's a really common misconception that inhaling is a process of, like, sucking air into the body.
Right, it's not a vacuum. It's really just an application of Boyle's law, manipulating volume and pressure.
Exactly. Inhalation is an active muscular process. The diaphragm, which is a dome-shaped muscle at the base of the chest, contracts and pulls downward, flattening out.
And at the same time, the intercostal muscles contract, pulling the rib cage up and out.
Right. This muscular action significantly increases the internal volume of the chest cavity. And as physics dictates, when volume increases in a closed space, pressure decreases.
So the pressure inside the lungs drops below the atmospheric pressure outside.
And because nature always seeks equilibrium, air just rushes down the trachea and into the alveoli simply to equalize that pressure gradient.
It's like a reverse vacuum cleaner. We don't actually suck air in, we just make the room bigger, and the air invites itself inside to equalize the pressure.
That is exactly what happens. And exhalation, under normal resting conditions, is largely just the passive elastic recoil of this system. The muscles relax, the volume decreases, the pressure rises above atmospheric levels, and the air is pushed back out.
But introducing this much highly branched, thin, wet tissue inside our bodies creates a unique physical hazard. We know that water molecules love to form hydrogen bonds with each other, creating significant surface tension.
Yes, they want to stick together.
So if you have millions of microscopic, wet, balloon-like alveoli constantly inflating and deflating, the surface tension of the water inside them should cause the walls to stick together and collapse entirely when you exhale, right?
It absolutely should. That phenomenon is called atelectasis, and it is prevented by another specific lipid structure called lung surfactant.
Kind of like the phospholipids in the cell membrane.
Very similar. Certain cells in the alveoli secrete this pulmonary surfactant, which is a mixture of phospholipids and proteins. It coats the inner lining of the alveoli, inserting itself between the water molecules.
Disrupting the hydrogen bonds.
Exactly. It drastically reduces the surface tension, which keeps those delicate air sacs propped open.
The biological engineering is just brilliant. But by opening up 75 square meters of delicate, single-cell thick tissue to the outside environment, we aren't just inviting in oxygen.
No, we are exposing our bloodstream to dust, pathogens, and particulate pollution.
The respiratory tract has physical defenses to manage this, though. The epithelial lining secretes mucus to trap incoming debris.
And embedded within this lining are cilia. They are these microscopic hair-like structures that beat in a synchronized rhythm.
They function as a mucociliary escalator, right? Just constantly sweeping the dirt and contaminated mucus upward to the back of the throat.
Yes, where it can be swallowed and naturally destroyed by stomach acid.
But the scale of modern environmental pollution is really overwhelming this fragile system. There is heavily documented epidemiological data linking air pollution, specifically in rapidly industrializing Asian cities, to a rising prevalence of severe respiratory issues like allergic asthma.
The primary culprits there are PM10 particulates. That's particulate matter that is 10 micrometers or smaller in diameter.
They are so tiny.
Because they're so incredibly minute, they easily bypass the mucus and the beating cilia. They travel deep into the terminal bronchioles and the alveoli.
And once they are down there, in the deepest part of the lung, those physical defenses are gone. The body has to rely on alveolar macrophages to patrol the area and engulf these foreign particles via phagocytosis.
But these macrophages evolved to digest organic matter, like bacteria, when they engulf inorganic PM10 particles like silica or heavy metals from exhaust. They literally cannot break them down.
So what happens to the macrophage?
They often rupture, and when they die, they release inflammatory cytokines into the surrounding tissue. This triggers a cascade of chronic inflammation and oxidative stress.
Which severely compromises that single-cell thick squamous epithelium.
Exactly. And that heavily correlates with the onset of allergic asthma.
So what does this all mean for us? From the single cell to the entire human body, life is entirely dependent on managing these selective borders. We saw how the phospholipid bilayer acts as a fluid dynamic bouncer at the club of the cell, relying on passive diffusion and active ATP-driven pumps to fight equilibrium.
And we looked at how water's pursuit of osmotic balance shapes everything from a bursting animal cell to the turgor pressure of a towering plant.
And it forces our amoeba to actively bail itself out. And we explored how complex organisms mathematically cheat the limits of diffusion by folding an immense, fragile, incredibly thin tennis court into our chests to hack Fick's law.
If we pull back from the biology for just a moment, I think it's worth pondering that the principles of Fick's law, surface area, concentration gradients, and barriers, they are universal physical rules.
They don't just apply to biology.
No, they describe movement in any system seeking equilibrium. Think about the thermal dynamics of your house in the winter. Heat naturally flows from the high concentration inside to the low concentration outside, completely dictated by the surface area of your walls and the thickness of the insulation barrier.
Oh, that is so true. Or even information flow. You could apply the mathematical concept of a concentration gradient to how quickly a viral video diffuses through a population constrained by algorithmic barriers.
Exactly. Whether it is molecular, thermal, or cultural, everything in the universe is driven by a fundamental physical need to balance out gradients. Life is simply the localized, highly organized resistance to that balance.
That is a wild thought to leave on. Thank you for bringing your curiosity and joining us on this deep dive. The next time you take a deep breath, feel the muscular drop and pressure in your chest, and just appreciate the millions of microscopic gradients actively working to keep you alive. We will catch you next time.